This episode is sponsored by Squarespace. Go to squarespace.com/microcosmos to get a free trial and 10% off your first purchase of a website or domain. If you're like a big Stentor fan, it's probably surprising to hear that what we are currently looking at is a Stentor. It looks more like a green cloud propped at just the right angle to look like maybe like a rabbit's head. But Stentors are famously built more like trumpets, not weird distorted bunnies. So, what happened here? Well, we did. Yes, what you are currently looking at is James, our master of microscopes, using a microscopy slide as a cutting board. This is not our usual approach to Stentor care. Usually, he gets his Stentors from a nearby park, brings them home, and pampers them with a drop or two of milk. Within a day, they grow so big that they are visible to the naked eye. Stentors are single-celled eukaryotes, so the fact that they can get so large has always made them quite striking to us, and they are always fun to watch. The cilia around their oral apparatus are always buzzing and gathering food, and the stripes lining their bodies can be filled with pigments that make these giants even more vivid against the rest of the microcosmos. And we've always been intrigued by the famous capacity for Stentors to regenerate. So, we wanted to spend today watching a Stentor put itself back together. Now, usually, we try to avoid hurting our microbial friends. In this case, though, we made an exception because, as we will see, Stentors can handle it. Our Stentor friend from the beginning of the episode was a casualty of this goal, and as we can see, it's still a ways away from looking like an actual Stentor. We can see a little more definition to the shape, but not by much. It turns out that slicing up the Stentor took a bit more work than we originally thought it would. At first, James tried to be precise and use a razor blade to take on individual Stentors in his sample. But with each movement of the blade, the surface tension of the water would move the Stentor away. So, instead, he went for a much less precise strategy, chopping away at the slide so that he ended up hitting a bunch of individual Stentors. Then he used a pipette to grab individual pieces of Stentors and transfer them over to a new drop of water so that he could investigate them more closely. Now, it might help to take a look at a healthy Stentor to remember exactly what they're supposed to look like. Obviously, things change from species to species, but there's the general trumpet shape we've mentioned before. And at the broader end of the Stentor is its oral apparatus, fringed with cilia buzzing around to help the Stentor gather food from its surroundings. So when we return to our regenerating buddy, it's starting to look like a Stentor that you might quickly scribble on a napkin. Perhaps when you're telling your friend about the really cool Journey to the Microcosmos video you just watched. And examining the other diced-up Stentors from James's samples shows us just how many different injuries they've sustained. There's headlessness. Then there's these. It's not even clear if we should be calling these Stentors anymore. They're fragments, remnants of something that was once a Stentor. And yet, these organisms are remarkably capable of repairing themselves even from these tiny fragments. Our Stentor friend, for example, is looking practically refreshed. As it turns through the microcosmos, the light reveals the striations down its body, and it looks like there was hardly a wound there at all. If we look closer at another Stentor in the midst of patching itself up, we can see a much better view of those stripes that make them up. In this Stentor, the parts without the stripes are the damaged part of the Stentor's cell membrane, and if you zoom in closer, you can see the pigment granules that make up those stripes. When you see just how many granules there are, not to mention how neat and orderly the pattern formed from them is, the feat that stentors are performing in their regeneration becomes even more incredible to comprehend. Like, sure, yes, it's incredible to see our stentor look more and more like a stentor from this distance, but it's almost like looking at a piece of art. You may not notice all the details that make it a masterpiece, but they are there and they are vital. Scientists have been studying stentor regeneration for decades, chopping them up, often with a bit more precision than we exhibited here, so that we can watch a stentor reassemble its oral apparatus or whatever body part they're interested in. Those experiments have taught us quite a bit about how stentors regenerate. For example, the primary requirement for regeneration is that the stentor fragment contains part of the macronucleus, the large nucleus that extends throughout the stentor, because the macronucleus contains many, many copies of the stentor's genome. So even with a tiny bit of the macronucleus, the stentor has all of the instructions it needs to remake itself. And this process can be done in a couple of hours. The molecular mechanisms that make stentors able to coordinate the many, many steps needed to rebuild themselves remain a mystery. But however mysterious, we can see those processes at work. Every time we return to our stentor, it's like it manages to look more and more like a stentor, even if we can't pinpoint all of the specific changes that refine that appearance. And this is where we will leave our friend, repaired and wandering around the microcosmos. Perhaps one day it'll run into another stentor, remade from another fragment of that original stentor. But by that point, I suppose, they will be two new entities entirely, like two ships passing in the night, not knowing that they were built with the scrap of the same old ship. Thank you for coming on this journey with us as we explore the unseen world that surrounds us. And thank you to Squarespace for sponsoring this episode. Squarespace gives people a powerful and beautiful online platform from which to create their website. So whether you're looking to create an online portfolio, start a blog, or open an online store, Squarespace can help you out. Maybe it's finally time to start that photography blog you've been thinking about. Well, Squarespace has the tools you need. They make it super simple to categorize, schedule, and share your blog posts. And once your blog takes off, you're going to want to connect with your audience. And guess what? Squarespace can help you out there as well. You can interact with your community on your Squarespace site through threaded comments and replies. And you can even generate extra revenue through members-only content. And if you decide you want to start selling prints of your fantastic photos, well, you already know what I'm going to say. Squarespace can help you there as well. Squarespace is already a powerful e-commerce platform, but you can even go the extra mile and add third-party extensions to help you manage inventory, streamline bookkeeping, and ship products across the world. And you can do all of this on one easy-to-use platform. Go to squarespace.com to sign up for a free trial, and when you're ready to launch, go to squarespace.com/microcosmos to save 10% off your first purchase of a website or domain. All the people on the screen right now, they are our Patreon patrons. Now, maybe some of them aren't going to be super happy about how we treated our stentors today, so I don't want to say that they are technically responsible for the content of this episode, but without them, this channel would not exist at all. So we are super grateful for them, and if you would like to become a patron of Journey to the Microcosmos, you can go to patreon.com/journeytomicro. If you want to see more from our master of microscopes, James Weiss, you can check out Jam and Germs on Instagram. And if you want to see more from us, there is probably a subscribe button somewhere nearby.